In the 19th century, unexplained forces routinely wiped out entire herds of livestock with anthrax, collapsed agricultural economies through sudden spoilage, and killed humans through cholera and rabies. The leading medical explanation for this widespread death was miasma, the belief that disease was caused by breathing in abstract bad air emanating from decaying matter. An even more dangerous assumption was the theory of spontaneous generation. This was the widely accepted idea that life, like maggots or disease-causing matter, simply materialized out of thin air whenever organic material began to rot. Humanity was fighting a war with zero intelligence on the enemy. Doctors and farmers couldn't stop the slaughter because the true rules of biology were hidden behind these abstract theories. Louis Pasteur entered this landscape as a chemist. His early career was defined not by medicine, but by the study of how atoms are physically arranged in crystals. This diagram represents his 1848 discovery involving tartaric acid. He found that identical chemical compounds can exist as mirrored versions of each other, structures that are chemically the same but physically oriented in different directions, like a left and right hand. This proved that the microscopic world was governed by specific physical geometry. If the building blocks of life had a concrete physical structure, then biological processes like fermentation and infection were likely caused by physical entities with their own unique shapes. This realization gave Pasteur the necessary logic to stop searching for abstract bad air and start searching for specific physical microscopic trespassers that could be seen, trapped, and studied. To prove these invisible trespassers were real, Pasteur had to physically trap them. To test spontaneous generation, Pasteur boiled broth in a flask with a long, curved neck. The open tube allowed air to flow inward. Airborne microbes were trapped by gravity in the lowest bend. Air passed, but microbes didn't, keeping the liquid perfectly clear. This proved microbes do not appear out of thin air. They are carried in the environment, and they must physically enter a host to cause change. By the 1860s, the French wine and beer industries were facing massive financial losses. A mysterious spoilage was turning vats of alcohol into vinegar. Pasteur investigated and found that while living yeast was responsible for normal fermentation, separate, unwanted bacteria were causing the spoilage. His solution was a targeted application of temperature. He discovered that briefly heating the liquids to between 50 and 60 degrees Celsius killed these specific spoilage microbes without altering the beverage's flavor. This process, called pasteurisation, gave humanity a repeatable method for neutralizing microscopic threats through physical control. Neutralizing a microbe in a bottle is useful, but building immunity in a living host requires a more sophisticated form of manipulation. The breakthrough came when a culture of chicken cholera bacteria was left sitting out on a desk over a holiday. The prolonged exposure to the environment weakened the pathogen. When Pasteur injected chickens with this weakened culture, they survived. Crucially, they were then immune when he later hit them with a fresh, lethal dose. He quickly applied this same methodology to livestock, creating a way to protect cattle and sheep from the devastating effects of anthrax. Pasteur discovered how to use the pathogen as a training tool for the biological immune system, turning a threat into a form of protection. In 1885, this logic was applied to save a human life. A 9-year-old boy named Joseph Meister had been mauled by a rabid dog, facing an almost certain death from rabies. Pasteur treated the boy using increasingly potent nerve inoculations harvested from infected rabbits, following his method of using weakened pathogens to build resistance. The boy survived. This success provided the first human evidence that artificial immunity could be induced, even against a highly lethal virus. It proved that instead of hoping to avoid an infection, medicine could preemptively train the human body to recognize and destroy a specific biological predator. The recognition of germs as physical realities meant that Pasteur's focus on sanitization and boiling quickly moved from the lab to the hospital. His work directly influenced surgeon Joseph Lister, who developed antiseptic surgery. This introduced a standard of cleanliness that significantly reduced fatal postoperative infections in hospitals worldwide. In 1887, the Pasteur Institute was founded in Paris, establishing a permanent center for the continued study of infectious diseases. Pasteur's work provided a repeatable protocol for modern public health: identify a physical pathogen, neutralize its spread, train the body to resist it.